Effect of the controlled delivery of chelating agent on the pulp tissue dissolution ability of fresh sodium hypochlorite solution by Natalia in 2023. Different forms of the salt of EDTA have been described, such as disodium, trisodium, and tetrasodium EDTA. Also, etidronate, which is a weak biocompatible chelator with pH varying from 10 to 12, it's used in combination with sodium hypochlorite, has been suggested to reduce formation of smear layer and reduce formation of accumulation of debris during root canal preparation. However, it requires longer time than EDTA to achieve the same result. At the same time, chelators like EDTA interact with sodium hypochlorite affect its tissue dissolution ability by reducing the active chlorine content. However, this effect was not observed with the etidronate and the salts of EDTA. So the purpose of this study was to evaluate the pulp tissue dissolution ability of reversed sodium hypochlorite when mixed with disodium, EDTA, and two different concentrations of etidronate, 9% and 18%. The pulp specimen obtained from 6 anterior teeth of bovine jaw were randomly distributed into 4 experimental groups and 2 control groups according to the irrigation protocol. Group 1 containing continuous delivery of 3% of sodium hypochlorite. Group 2 continuous delivery of sodium hypochlorite 3% plus alternate delivery of 17% disodium EDTA. Group 3 continuous delivery of 3% sodium hypochlorite plus alternative delivery of 18% etidronate. Group 4 continuous delivery of 3% sodium hypochlorite plus alternate delivery of 9% etidronate. Negative control distilled water. Positive control continuous delivery of 6% sodium hypochlorite. Each sample was introduced into a centrifuge tube that contained inner filter. The filter was placed in 50 milliliter plastic tube. Both distilled water in the negative control and sodium hypochlorite in the positive control were continuously delivered with a pump, and 24 9mm needle under continuous controlled rate, 1 milliliter per minute. A second pump was and a second needle alternatively delivered either EDTA or etidronate at the same rate with 30-second programmed interval. The delivery of sodium hypochlorite with the first pump was not interrupted with the second pump when the second pump was activated. Both needles were fixed to the cap of the plastic tube, allowing individual solution not to contact before the test. The pumps were stopped after 2 minutes of application of the irrigant solution. Samples were then washed with 2 milliliter distilled water for 30 seconds to neutralize the effect of the different solution. Bottled dry again and reweighted to evaluate the remnant tissue. The same procedure was repeated after 5 and 10 minutes, T5 and T10. Regarding the available chlorine and the pH and the temperature, 3 samples in each group were randomly selected for further registration of free available chlorine pH at T0 and T10. A standard iodine thiosulfide titration method was used to calculate the available chlorine content in the sodium solution at T0 and T10 in all study groups, and initial and final pH of each solution were registered with calibrated pH meter. Figure 1 demonstrates the model of the tissue dissolution. Regarding the results, values of available chlorine and pH for each group at T0 and T10 are as follows. Available chlorine did not change in this supernatant for both positive control and group 1. However, a reduction in the available chlorine was observed after 10 minutes in the rest of the groups. Other reactions were also observed in the supernatant of group 2, decreased in initial pH, the presence of bubbles due to gas formation, and increase in the temperature, 35 Celsius degree. This exothermic reaction was not observed in the rest of the groups. Moreover, at the end of the experiment, the filter containing the pulp specimen presented salt precipitate. The current study demonstrates that alternative delivery of EDTA and etidronate under controlled flow did not affect the pulp tissue dissolution ability. Refreshed sodium hypochlorite. Further research should be confirmed its effect in the presence of dentine and preferably in human root canals. With the limitation of this study, it can be concluded that the controlled delivery of EDTA and isidronate did not alter the tissue dissolution ability of sodium hypochlorite when first solutions were mixed in the root canal.